Okay, sorry about that. I didn't foresee the fire alarm going off. Right. Okay, so we were just discussing the 5 different families of G protein coupled receptors, and we got to the 5th and final family, which was the frizzled /taste2 receptor family. Okay, and this is named after the 2 main members of this family, which are the frizzled receptor and the taste2 receptor. Now, these receptors will have reasonably large amino terminal domains, and then they'll have, of course, the 7 conserved membrane-spanning alpha helices, like so, okay, culminating with the carboxylic acid tail here, and here's the amino terminus up here, and then basically their ligand binds extracellularly, okay, so the ligand might bind here. So it's going to bind to the amino terminus domain. Okay, so this is kind of the family with everything that doesn't fit into any of the other families. Okay, so the ligand doesn't bind to the transmembrane domain, so they're not in the rhodopsin family. It doesn't get wedged in between the transmembrane domain and the amino terminal domain, so we're not talking about the secretin family. There's no venous flytrap domain, so we're not talking about the glutamate family. It's not a component of the extracellular matrix, so we're not talking about the adhesion family. What's left is the frizzled /taste2 family. Okay, and clearly the 2 main members are frizzled and taste2, and frizzled is involved in the Wnt beta-catenin pathway, so its ligand is Wnt, and taste2, the ligand for taste2 is molecules of bitter tasting molecules, and it's involved in the gustatory system. Okay, right. So those are the 5 different families of G protein coupled receptors. What we now want to talk about is how they actually produce signaling cascades, i.e. how they actually interact with heterotrimeric G proteins. Okay, right. So let's now talk about heterotrimeric G proteins. So we'll now go back to talking about just a generic G protein coupled receptor, rather than it being in any of the 5 families. And what we know, the generic G protein coupled receptor has these 7 membrane-spanning alpha helices, with an amino terminal domain extracellularly and a carboxylic acid tail intracellularly. Right, so basically you start off with the resting receptor here. And basically at the moment it's not activating any G proteins, okay? But the G proteins that it can interact with are heterotrimeric G proteins. So let's talk about the structure of heterotrimeric G proteins. So basically heterotrimeric G proteins, now write the name out in full, okay? So heterotrimeric G proteins. Basically, the hetero means different, trimeric means 3-membered. G proteins refers to the fact that they are guanine nucleotide regulatory proteins, or rather, I've missed a word out there, guanine nucleotide binding regulatory proteins. That's what the G is short for. It's short for guanine nucleotide binding regulatory, okay? So just for short, G proteins, okay? And heterotrimeric G proteins consist of 3 separate subunits bound together. At least they do in the inactive state, okay? So let me explain what I mean by that. So basically, here we have the alpha subunit of the heterotrimeric G protein. So all heterotrimeric G proteins consist of 3 subunits, an alpha subunit, a beta subunit, and a gamma subunit. And the alpha subunit is quite special. It can be in 2 states, okay? There are 2 states it can be in. It can either be in an off state, and in the off state it has guanosine diphosphate bound to it, or it can be in the on state where it has guanosine triphosphate bound to it, okay? So let's say that our alpha subunit currently has guanosine diphosphate bound to it, and therefore is in the off state, okay? So I'll colour this in red here, okay? So basically, the other thing that I need to tell you about for the alpha subunit is that it's got this special lipid moiety stuck onto it, okay? So basically, absolutely all alpha subunits of heterotrimeric G proteins get a special molecule stuck onto the side of them, okay? And this molecule is either a myristic acid molecule, okay? And myristic acid is the old name for something that would now be called tetradecanoic acid. And although tetradecanoic acid might be a bit of a mouthful, it's useful because it tells you exactly what the structure of this molecule is. It tells you that we're just dealing with a 14-carbon, fully saturated carboxylic acid, so there's nothing complicated with regards to myristic acid. It's just a 14-carbon long molecule with a carboxylic acid group right at one end and then hydrogens everywhere else. Okay? Then there's another option that it could be, which is palmitic acid, and again palmitic acid is the old name, and it's the name that's still very prevalent in biochemistry, but the name that chemists would now use for palmitic acid is to call it instead hexadecanoic acid. And again, this tells you exactly what the structure of this molecule we're dealing with is. It tells you that we're dealing with a 16-carbon fully saturated carboxylic acid. So basically, every single alpha subunit gets one of these two carboxylic acids added onto it, or some of them even get both of them added onto them. Okay, and what's the significance of this? Well, it basically means that the alpha subunit has a really long chain carboxylic acid group sticking off the side of it, okay? And the tails of these molecules are extremely hydrophobic. So what can happen is these tails can implant into the inner leaflet of the phospholipid bilayer, which has a lot of hydrophobic molecules in, basically. So you can implant this hydrophobic tail into the inner leaflet of the phospholipid bilayer, and that anchors the whole alpha subunit just underneath the plasma membrane, basically. And just to make sure that we're all on the same page, the inner leaflet of the phospholipid bilayer is the name for the inner layer of phospholipids of the phospholipid bilayer. So this is the inner leaflet, okay? So basically what this does is it attaches the alpha subunit to the base of the plasma membrane, basically, and that's how these alpha subunits stay near the plasma membrane, near to the G protein coupled receptor here. Okay, right. Now, that's not the only subunit of a heterotrimeric G protein. When the alpha subunit is in the off state, it can associate with two other subunits of heterotrimeric G proteins, okay? And these two never break apart, okay? So I'll draw these two here. So there are the other two subunits of the heterotrimeric G protein. After all, it is a free-membered complex. They never break apart, so they're very faithful to one another. So you have the beta subunit and then also the gamma subunit. So they're named after the first three letters of the Greek alphabet: alpha, beta, gamma. So we'll have beta in blue here, okay, and then we'll have gamma in green. Okay, so here we go, alpha, beta, and then gamma. Now, the gamma subunit also has a very hydrophobic lipid moiety stuck onto it. Now, it's a slightly more complicated lipid moiety that the gamma subunit gets added onto it. It gets something called a prenyl group added onto it, which will either be farnesyl or geranyl. Okay, now if you don't know what those are, don't worry. All you need to understand is that you've got a very long chain hydrophobic molecule stuck off the side of the gamma subunit, and this is implanting itself into the inner leaflet of the phospholipid bilayer, and that means that the beta-gamma complex, as it's called, because the beta and the gamma subunit never break apart, it makes sense to talk about them as one, basically, the beta-gamma complex. Okay, this attaches the beta-gamma complex to the inner leaflet of the phospholipid bilayer, so again, they are floating around underneath the plasma membrane. Right, so when the alpha subunit has GDP bound to it and is in the off state, the beta-gamma complex can associate with the alpha subunit. Okay, right, now, when the G protein coupled receptor over here gets its ligand bound to it, what will happen is the G protein coupled receptor will change conformation, okay? And this change in conformation will be transferred down to these 3 intracellular loops here. So this is intracellular loop 1, this is intracellular loop 2, and this is intracellular loop 3. And these 3 intracellular loops will change conformation, and they will make available a binding site for the alpha subunit of the heterotrimeric G protein. So it will change conformation to produce a binding site for the alpha subunit of the heterotrimeric G protein. So I want to stress again that it's only a binding site for the alpha subunit. However, the alpha subunit can only actually bind to this if it's got the beta-gamma subunit with it. Okay? So without the beta-gamma complex, the alpha subunit alone cannot bind to this G protein coupled receptor, basically. So although, yes, it only binds to the alpha subunit, not the beta-gamma complex, you have to have the beta-gamma complex in tow to actually be able to bind there. Okay, now, there are different alpha subunits, and we'll come to this in a moment, and different G protein coupled receptors will bind to different alpha subunits, basically. So they won't bind to absolutely any old alpha subunit. They'll have preference for different alpha subunits. Okay, but we'll come to the diversity of G protein coupled receptors in a moment. I just now want to discuss the G protein cycle first, and then we'll come to the different alpha subunits, because that's going to be something that's quite a lot of information to take in, and it's also something that's actually very important for us to understand the function of cholera and Pertussis toxin. So we need to have it fresh in our mind when we actually come on to cholera and Pertussis toxin. So I'll leave that until last. Okay, right. So basically, where were we? The G protein coupled receptor cycle then. Okay, so basically what's going to happen is the G protein coupled receptor is going to change conformation when the ligand binds, and it will now make available a binding site for certain alpha subunits. So let's draw this, and I think we'll try and squash it in down here. Okay, so to make the drawing nicer, I'm going to flip the G protein coupled receptor around and put the carboxylic acid tail this side. So I'm just rotating it around 180 degrees. It'll just make the picture look nicer. Okay, it's the exact same receptor though in principle. So we'll have HOOC this time, and then we'll have the amino terminus over there. Okay, so now I can draw this alpha subunit nicely coming in and binding to the intracellular portion of the receptor. Okay, so here it is, and it still has its lipid moiety attaching it to the plasma membrane. How I'm going to show this is another matter. Okay, there we'll go, that can be it. In fact, I'm going to colour that lipid moiety in. So the myristoyl or palmitoyl group, I'm going to now colour in pink. So there it is. Okay, and in then comes the alpha subunit, so I'll colour this in red. And at the moment it's still got GDP bound to it. Remember, the beta-gamma complex will not bind to the alpha subunit unless it's got GDP bound to it. Okay, so here is GDP. Okay, and then we've also got this beta-gamma complex here, which is attached to the alpha subunit. Even though it doesn't look as though it is from my picture, it should be attached to that alpha subunit. Okay, right, so we'll colour this in. And basically, I will stress again that the intracellular aspect of the G protein coupled receptor will only bind to the alpha subunit, and it will only bind to it if it is in association with the beta-gamma complex. Okay, now, what then happens is when the alpha subunit is bound to the intracellular aspect of the G protein coupled receptor, what happens is the G protein coupled receptor catalyzes the throwing out of the guanosine diphosphate and the bringing in of a guanosine triphosphate molecule from the cytoplasm, which will bind to the guanine nucleotide binding site of the alpha subunit. So basically you take away the GDP and you bring in GTP, so the alpha subunit now changes conformation and is now in the on state. Now as soon as it binds the GTP and changes conformation, it no longer binds to the beta-gamma subunit, so that goes off. So the beta-gamma complex will wander off, basically. It abandons the alpha subunit. Okay, and then when the alpha subunit no longer has the beta-gamma subunit with it, then the alpha subunit can no longer bind to the intracellular aspect of the G protein coupled receptor. So it detaches from the G protein coupled receptor, and the overall result of this then is that we have the beta-gamma complex wandering off, and the alpha subunit with GTP bound to it also wandering off. Okay, so let me draw a picture of this. So here is our plasma membrane here, and so far what we've got is this alpha subunit which still has its myristoyl or palmitoyl group attached to it, and that's anchoring it in the inner leaflet of the phospholipid bilayer, and then it has guanosine triphosphate bound to it. Okay, and then you also have the beta-gamma complex over here, again, which has this prenyl group that anchors it into the inner leaflet of the phospholipid bilayer. Okay, so let's show the alpha subunit here in red, like so, and then we'll show the beta-gamma in the conventional colours of blue and green. Okay, so here's the beta in blue and the gamma in green. Then what will happen is both of these go off and have downstream effects, basically. So both of them will have targets that will generally also be in the plasma membrane, or at least attached to the plasma membrane. So let's draw a target for the beta-gamma complex here. So here is target, a target for the beta-gamma complex. Okay, and then we'll have some target for the alpha subunit, which has GTP bound as well. Okay, so this is something that's quite important to understand. People generally forget about the beta-gamma complex, but its role in physiology, well, its role in microphysiology and molecular biology, that's the word, it's becoming more and more important, and it's becoming acknowledged how important the beta-gamma complex is in signaling. Okay, we're seeing more and more examples of where the beta-gamma complex is doing stuff that the alpha subunit doesn't, basically. Okay, so both of them have signaling roles. So this one will go off and bind to a target, this one will bind to a target, they'll both do stuff. Okay, now how is the signal then terminated? Well, basically, when the alpha subunit binds to its target, what happens is this target activates the alpha subunit. Okay, and this is where it becomes important to understand that the alpha subunit is actually an enzyme. Okay, it's a GTPase. It's an enzyme which can chop the gamma phosphate group of GTP and convert it back to GDP. So it catalyzes the conversion of GTP to GDP. Now, it actually has a GTP molecule bound to it, so it can actually catalyze the conversion of GTP into GDP. Okay, now, usually it doesn't do that, okay, but once it has bound to the target, well, it does do that, but very slowly. Okay, so its usual GTPase activity is very slow. However, once it's bound to the target will activate the GTPase activity of the alpha subunit, and it will suddenly speed up the speed at which it catalyzes this reaction. Okay, the rate at which it catalyzes this reaction, that's the word I was looking for. And it will convert the GTP, therefore, to GDP, and it will therefore inactivate itself. So what will happen is the alpha subunit will effectively turn itself off as soon as it starts actually doing some signaling. So as soon as it starts binding to targets, those targets will activate it to switch itself off, basically. Okay, so now we've got the alpha subunit with GDP bound to it, and as soon as the alpha subunit turns the GTP back to GDP, then it will cleave away from its target, okay? So the target only wants to bind the alpha GTP subunit. Once it's an alpha GDP, it's not interested in it anymore, and the alpha GDP will then go back it'll go back into the free inner leaflet of the phospholipid bilayer, and it'll wander around a bit until it finds the beta-gamma complex, and then it will bind to the beta-gamma complex again and wrench it off the beta-gamma complex's target. Okay, so the two will then reassociate, and you're back to the beginning, basically, as though nothing ever happened. Right, okay, so that cycle whereby you go from the inactive heterotrimeric G protein and then you return all the way back to the inactive heterotrimeric G protein, that is what's known as the G protein cycle. Okay, right, so we'll call it there for this video, and in the next video what we'll do is get some idea of the diversity of heterotrimeric G proteins. So we'll look at the different alpha subunits, the different beta subunits, and then the different gamma subunits, and we will then be in a position to look at the function of cholera toxin and pertussis toxin.